Morning students. Today let us discuss the structure of proteins. Proteins are polymers of amino acid monomers. The amino acids are linked together to form a chain called peptide chain. The peptide chain has a variable length and it depends on the number of amino acids linked. The amino acids of proteins are called residues. In this picture you can see the amino acids, peptide chain and protein molecule. In this picture, you can observe the molecular formula of protein. The term protein is derived from Greek proteo, means primary or holding first place. The term protein was first proposed by Berzelius. Protein is the essential constituents of living cells. Proteins made up of 12% of the protoplasm. They are the body builders. They contain carbon, nitrogen, hydrogen, oxygen and sometimes sulfur. They are constructed largely of amino acids. Structure of protein. Lindström-Lang suggests 4 types of structural organization of proteins. They are primary structure, secondary structure, tertiary structure and quaternary structure. Now let us see the primary structure. The primary structure of protein is defined as the linear sequence of amino acid residues making up its polypeptide chain. The protein may be formed of one or more polypeptide chains. The amino acids are arranged in specific sequence in the polypeptide chain. The amino acid residues are linked by peptide bonds. The peptide bond is formed between the carboxyl group of one amino acid and the amino group of adjacent amino acid. You can see this in the picture. Sometimes the adjacent polypeptide chains are linked by disulfide bonds. Many structural proteins which form fibers are linear and unfolded types. Example, silk fibroin. The peptide bond is a repeating unit. 2 amino acids give a polypeptide, 3 amino acids give a tripeptide, 4 amino acids give a tetrapeptide and so on. Less than 10 amino acids form an oligopeptide. More than 10 amino acids form a polypeptide. Each polypeptide chain of any length has its one end an N-terminal amino acid containing a free amino group and at the other end C-terminal amino acid containing a free carboxyl group. The amino acids in a polypeptide chain are numbered from N-terminal end. Thus the primary structure has the following salient features. Primary structure refers to the linear sequence of amino acid residues. The proteins are linear and unfolded. The protein is formed of one or more polypeptide chains. The amino acid residues are linked by repeating peptide bonds. The adjacent polypeptide chains are linked by disulfide bonds. Most of the structural proteins which are in the form of fibers exhibit primary structure. Example, fibroin of silk. Secondary structure. Secondary structure refers to the helical nature of the proteins. The secondary structure is derived from the primary structure by the formation of hydrogen bond interactions between amino group residues fairly close to one another. This leads to the folding of the polypeptide chain into a helix. The intramolecular hydrogen bonds occur between an amide nitrogen and the carbonyl oxygen of the 4th amino acid residue of the polypeptide chain. In addition, disulfide bonds also occur either in the same chain or between polypeptide chains. By configuration, the secondary structure is classified into 2 types, namely helical structure and plated sheets. Helical structure. The polypeptide chain is coiled as a rope wound around a cylinder. The helical structure of protein is formed by the hydrogen bonds between peptide groups within the same polypeptide chain. The most common form of coiling is the right-handed alpha helix. Only right-handed alpha helix occurs in nature. The alpha helical structural model was proposed by Pauling and Corey in 1951. The main features of alpha helix are: alpha helix is formed by a series of amino acid residues woven into a spiral chain. The alpha helix is so named because of the mobility of alpha carbon atoms. The helical chain has a screw-type symmetry. A hydrogen bond is formed between the peptide groups CO and NH of every first and fourth amino acid residues. The regular appearance of hydrogen bonds between every first and fourth peptide groups determines the regularity of turns in the polypeptide chain. The shape of the helix is maintained by hydrogen bonds. Each complete coil contains 3.6 amino acid residues. Second type, plated sheet structure of beta structure. It is a secondary structure formed from hydrogen bonding between two peptide chains. This bonding leads to the formation of sheets of parallel chains in the form of plated sheets. In plated sheet structure, the chains may be parallel or anti-parallel. In a parallel chain, plated sheet structure, the N-terminal end of the polypeptide chains point in the same direction. Example, keratin. In anti-parallel chain plated sheet structure, the N-terminal ends of the polypeptide chains point in opposite directions. Example, fibroin of silk. Tertiary structure. The tertiary structure of protein is more complex than the secondary structure. Tertiary structure is exhibited by proteins having only one polypeptide chain. The tertiary structure is attained by globular proteins. In such cases, the secondary helical structure is further folded to form a globular ellipsoidal shape. The folding is established by the appearance of more disulfide bonds as well as hydrogen bonds, ionic bonds and hydrophobic bonds. Example, myoglobin, ribonuclease, chymotrypsin, cytochrome C, etc. They are exist in tertiary structure. Myoglobin is abundant in muscles. It contains single polypeptide chain of 153 amino acids and a single iron porphyrin heme group. The tertiary structure of myoglobin has a sausage-like outline. The backbone of the molecule is made up of 8 almost straight helical segments alternated by non-helical bends which twist to produce the characteristic foldings. This you can observe in the picture. The longest helical segment has 23 amino acids and the shortest segment has 7 amino acids. All the helical segments are of alpha type and right-handed, there being no beta structure. Nearly 80% of amino acids remain on the helical regions and the 20% are distributed over the non-helical areas. Ribonuclease is another globular protein with tertiary structure. It is made up of single polypeptide chain containing 124 amino acids with 4 disulfide bonds. Quaternary structure. 2 or more polypeptide chains associate together to produce a quaternary structure. It is exhibited by proteins containing more than one polypeptide chains. Quaternary structure is formed by the combination of primary, secondary and tertiary structures.